Claire Robinson's book, GMO Myths and Truths, A Citizen's Guide to Genetically Modified Crops and Foods, dives into a topic that has sparked intense debate for decades: genetically modified organisms, or GMOs. The journey of GMO myths and truths began in 2010, when Robinson and her co-authors noticed a troubling trend. Despite a growing body of scientific evidence pointing to potential risks and actual harm from GMOs, the public was being told that there was no science behind the case against genetically modifying our food supply. This was a claim she and her co-authors knew to be untrue, having followed the scientific debate since the early 1990s. They were also concerned about the inflated claims being made for GM crops, such as promises of increased sustainability, higher yields, reduced pesticide use, and more nutritious foods. These claims, as the authors discovered, were at best questionable, and at worst blatantly false. One of the most persistent myths about GMOs is that genetic engineering is just an extension of natural breeding, and therefore no more risky. However, the truth is that genetic engineering is radically different from natural breeding and poses unique risks. Genetic engineers argue that GM is more precise, allowing genes for desired traits to be inserted with minimal unexpected effects. But the reality is that GM involves a random insertion of an artificially constructed foreign gene unit, leading to unpredictable changes in the DNA, proteins, and biochemical composition of the resulting GM plant. The process of creating a GM crop is anything but natural. It involves isolating genes of interest, cutting and splicing them to create a GM gene cassette, and then inserting this cassette into cultured plant cells. This is done using methods like biolistics, or infection, with Agrobacterium tumefaciens bacteria, both of which are random processes that genetic engineers cannot fully control. The result is a highly imprecise and unpredictable process. Plant cells that have successfully incorporated the GM gene cassette are selected using marker genes, often conferring resistance to antibiotics or herbicides. These cells are then stimulated to grow into small GM plants. The GM plants are tested to identify those expressing the GM genes at desired levels and locations. Only a few out of hundreds or thousands of plants may meet these criteria and become candidates for commercialization. This process is highly mutagenic, causing potentially damaging mutations throughout the plant cell's DNA. Despite claims that harmful mutations are eliminated during development, subtle changes in the plant's biochemistry can still occur, leading to unexpected and potentially harmful effects. The regulation of GM foods is another area where the truth is often obscured. The claim that GM foods are extensively tested and strictly regulated is misleading. In reality, they are primarily tested for safety by the companies that stand to profit from their sale, and these tests are often inadequate. The regulatory process relies heavily on industry studies, which are inherently biased. For example, the U.S. Food and Drug Administration, FDA, has allowed GM foods onto the market despite warnings from its own scientists that genetic engineering poses unique risks. The FDA's presumption that GM foods are generally recognized as safe, GRAS, is not based on scientific consensus. In fact, there is no scientific consensus on the safety of GM foods, as evidenced by statements from expert panels and scientists worldwide. The concept of substantial equivalence is often used to argue that GM foods are as safe as their non-GM counterparts. However, this concept is fundamentally flawed. It assumes that if a GMO contains similar amounts of basic components like protein, fat, and carbohydrate as its non-GM counterpart, then it is substantially equivalent. This approach ignores the complexity of genetic modification and the potential for unintended changes in the plant's biochemistry. Studies have shown that GM crops often differ significantly in nutrient levels and protein types from their non-GM counterparts. These differences can impact allergenicity, toxicity, and nutritional value. Yet regulators often dismiss these differences as not being biologically relevant without providing a clear definition of what constitutes biological relevance. The promise of increased yields and reduced pesticide use is another area where GM crops have fallen short. While some GM crops have shown short-term benefits, long-term studies and real-world experience tell a different story. For example, The widespread adoption of GM herbicide-tolerant crops has led to the rapid spread of herbicide-resistant superweeds, resulting in increased herbicide use, not less. Similarly, GMBT crops, designed to produce their own insecticides, have not eliminated the need for chemical insecticides. In fact, as insects become resistant to the BT toxins, farmers often return to spraying chemical insecticides. Additionally, the use of toxic neonicotinoid insecticidal seed treatments has increased on both GM and non-GM crops, further debunking the claim that GM crops reduce pesticide use. The health impact of GM foods is another contentious issue. While proponents argue that GM foods are safe, a growing body of scientific evidence suggests otherwise. Studies have found potential signs of toxicity and actual toxic and allergenic effects in laboratory and farm animals fed GMOs. For example, rats fed GMBT maize showed differences in organ and body weights and blood biochemistry, and histopathological investigations revealed toxic effects in multiple organs. Despite these findings, long-term and multi-generational studies are rare, and those that do exist are often dismissed by industry and regulators as not biologically significant. This lack of rigorous, long-term research makes it difficult to definitively assess the safety of GM foods. The environmental impact of GM crops is also a cause for concern. The adoption of GM herbicide-tolerant crops has led to increased herbicide use, contributing to the spread of herbicide-resistant weeds and the degradation of soil health. The chemical treadmill of relying on increasingly toxic herbicides is unsustainable and poses risks to both the environment and human health. Moreover, the claim that GM crops can coexist with non-GM and organic crops is misleading. Experience has shown that once GM crops are introduced, contamination of non-GM and organic crops is inevitable, removing choice from farmers and consumers. In conclusion, the evidence presented in GMO Myths and Truths challenges the narrative that GM crops are the solution to the world's food and environmental challenges. While GM technology may have a role to play in certain contexts, it is clear that the promises made by GMO proponents have not been realized. The reality is that GM crops have not delivered on their promises of increased yields, reduced pesticide use, or improved nutrition. Instead, they have introduced new challenges and risks. As we move forward, it is crucial to critically evaluate the claims made about GM crops and to consider the broader implications of relying on genetic engineering as a solution to complex problems. The future of agriculture lies in sustainable, ecologically sound practices that prioritize the health of both people and the planet. This has been a BrightLearn video from BrightLearn.ai on the book GMO Myths and Truths, A Citizen's Guide to Genetically Modified Crops and Foods by Claire Robinson. Visit BrightLearn.ai for more fascinating videos like this one, and visit naturalnews.com for full editorial coverage and breaking news on critical stories that keep you informed and aware of what's really going on.